Hey, it's Peter here with my FSHD. And well, Happy New Year 2026. Here we are, January 2026. God, I can't remember last time we were on. I guess we had Brad, our angry dad, on last time. I think that was before Thanksgiving. So happy Thanksgiving, happy Hanukkah, and Merry Christmas, everybody. When you don't hear from us, well, it's because we're really busy. And that's actually a good thing. Got a lot going on, writing grants, writing papers. Oh my God, are we writing papers? But the space is always moving. And one of the things I do actually like to hear from y'all, and most of the time, not everybody's so patient and nice, but most of you are wonderful, and I do love hearing. And thank you for all the Happy New Year's and well wishes. I really do appreciate it. I pass everything positive onto the lab, and but it's good to hear a lot of comments that you're getting really sciencey. Towards the end of last year, really, really sciencey, more a little over everyone's head, kind of losing everybody. And I'm in kind of a tough position. You got to remember, as a researcher, now that I understand that other researchers listen, NIH listens, companies listen, my competitors listens, my colleagues listen, foundations listen. I really just was setting this up to talk to patients, talk to you all and your families, and kind of bring things to you. So it really kind of start digging in deeper to things. And I've been thinking about what to do, and thought, maybe we'll kind of get back to our roots. And while I do want to be able to explain the science unbiasedly, now remember, when I'm going to be explaining to you, I have no affiliation with these companies. I'm not getting paid to endorse anybody. Directly or indirectly, and we just give you our honest truth. But I do want you to understand the science, and sometimes, especially if I'm not so enthusiastic about somebody's science, I really feel the need that I have to make the case and explain why. That I'm not just coming out saying, "Hey, I don't like it," gut feeling. So I have to explain. So sometimes we get a little sciencey, but today we're gonna try a little bit different because the more sciencey I get, the more work I make for myself. And it's really hard to make these presentations. Now we're doing video, so I'm going to try something a little different. We'll see how it goes. Let me know. We're going to be shorter also. I'm going to do things a little shorter. So the big news, of course, is that Arsenal's in first place in the Premier League with 17 games to go. So we'll blow it. Now, the big news, right? There's just been a whole lot of news in the FSHD therapeutic space, and a lot of people keep asking for my comments on it. People ask me routinely if I recommend that they join a certain trial or do not join a certain trial. I really don't like to do that so much. I'm happy to explain what the trials are about and explain press releases. Love to explain press releases, and that's kind of what we're doing today. So since I'm getting too sciencey for some of y'all, one of the things I'm going to do is just kind of pull from the layman science literature that's out there online, and use that, not really make presentations. So I'm going to start with something really important, because we're going to be talking about Epicrispr, their recent news press release, and their clinical trial that's phase 1/2. And then we'll be talking about the stem cell therapy trial that's out there being run at Stanford in John Day's clinic, and the Scholar Rock trial, which I'm actually very familiar with. Scholar Rock, known about them for quite some time, and I just kind of give you some kind of comments on those, and some opinions on what's going on. Okay, but first I want to start with something very important to remind people of a very important phenomenon. I hope you all can see the screen here. It's something called the placebo effect. Okay. So what's a placebo control? Well, if you have a research group like here, you have all these little blue dudes in a clinical trial. You split them, sometimes 1-to-1, sometimes 2-to-1, drug to, I think the Avidity phase 1/2 trial was 2-to-1. Phase 3 is 1-to-1, I believe, with one set gets the drug. The other set gets treated exactly the same, but they don't get the drug. Okay. Then you can also have crossover studies where, like for a gene therapy trial, what you might do is one set of, half the patients might come in and get an injection and get the drug, then a year later, and the other set gets an injection that does not have the gene therapy. And then a year later, you switch over and they both get a second injection, but of course you can't inject virus twice. So the ones that got the gene therapy the first time just get the saline the second time. The ones that got the saline get the gene therapy the second time. And that's a crossover. And then you can actually see if you get improvement in the placebo group when you go on the drug. So you can actually test two things. And so that's often how gene therapy trials are done. And so that way, and the great thing about eventually everybody being on drug is that when you have open label extension, the placebo group goes up to the drug group, and you can actually see then the improvement of the placebo group, right? And so it's a great benefit for people on trial when there's open label extensions, because everybody knows they're eventually going to be on drug. But at the time that you're in the trial, you don't know if you're on drug. It's 2-to-1 or 1-to-1 drug to placebo, but you actually don't know, right? Of course, you're hoping you're on the drug. And of course, you're telling the doc, hey, give me the good stuff. Normally, the person administering the drug is blinded to whether or not they couldn't give you the good drug if they wanted to, right? They have to give you what they get, right? It's blinded to everybody. But what the placebo effect is that a lot of people in this group that do not get the drug get benefit and report benefit and even have measurable benefit, depending on what you're measuring, okay? And so you actually get real improvement after taking a fake treatment. And so why would that be? Well, let's actually look at this. So pulled this from the Cleveland Clinic. What's the placebo effect? Why it happens, essentially. So it's the phenomenon is when your high expectations can influence how well a medication or procedure works. Basically, just by being in a clinical trial, your mood improves. Everything kind of like improves with you because you're doing something about the disease. And you know what? You might be on the drug, and you can convince yourself you're on the drug. You don't really know. And actually, I remember in the Fulcrum trial, I knew some patients that were like dissolving their pill in water to see if they could see and determine whether or not they were on drug or not. Instead of just taking their pill. That's actually a bad thing to do, by the way. Just take your pill, whatever it is. And, it's kind of your duty if you're in one of the trial to follow what they want you to do. Typically, what they want you to do in a trial is not change your lifestyle, not change anything. Just take the pill and go through and keep going. Yet, it's interesting. The Friends of FSH Research Gala is coming up, and I highly recommend everybody go. It's fantastic. Terry Colella, George Shaw, Rick Colella, the whole gang, Bradley Baxter, the whole gang up there at Friends of FSH Research does a great night, great auction, great everything. But, I was up there a few years ago, and I was talking with someone who was in the Fulcrum trial. He's like, it was life-changing. It was life-changing for him. He's like, now he's able to go out and exercise. He's able to do all these things. He's doing everything. He's like, oh, yeah, I'm walking all the time. I'm riding a bike. It's just been amazing. Now, he's currently in the trial, and he's doing all these things that he couldn't previously do. But he actually didn't know whether he was on the drug or not. I must be on the drug. I feel great. Maybe you're not. In fact, maybe that's screwing up. And actually, other people, I told a number of that one's really stood out. But there are others have talking to patients that they must be on the drug because they're more active during the day. Well, maybe you are, maybe you aren't. But why do they assume they must be? Well, high expectations influence how well a procedure works. This is actually something that's real, right? It says the mind and body connection is very powerful, right? That you can actually feel better when you're hurting, right? So what is this? It says they don't do anything physiologically speaking, but you can actually cause improvement based on your state of mind. You're trying harder. You're more willing to exercise. And what do we know with FSHD? We know that exercise improves health. We know getting out there and doing things gets better, right? All of you that are in the FSHD Supplements, Nutrition, and Peer Support Face group, the only Facebook group I actually recommend, run by Tamara Gottlieb and Emily Ka and the gang. It's fantastic, right? It's you can do things. So now if you're suddenly more willing to actually do these things, and then when you actually get measurements for how you're doing in the trial, your reachable workspace, your walking, your 6-minute walk, you're timed up and go, are you trying harder? Well, it's an interesting thing because you test the effectiveness of medical interventions in clinical trials, right? One group gets the medicine, one the placebo, just like we told you. And then you compare the two. And it's very often that both groups will see improvement. Just the act of taking a pill or getting a shot makes people feel better. Okay? And so why does it happen? Well, the stronger you build up a person's expectations and belief in their treatment, the more it can actually influence the outcome. Okay? That's actually, it's a real thing. If you think it'll work, you want it to work, So in a way, you subconsciously talk yourself into believing it's working. Okay. So it's what more, it actually, your body can actually have real physiological responses. Okay. So there you go. So now there's the other thing. There's also the opposite effect. Okay. It's called the nocebo effect. Okay. Negative expectations leading to a medical intervention can lead to worse outcome. For example, if you know that you're not on the drug and you know you're gonna just get worse, like in a natural history study, so you're participating in a study, but you're expected to get worse, you may go ahead and get worse. You're not likely to get better because you know you're not on a drug. Okay, so why do I bring these two things up? Okay, I'm gonna get to another one really quick. This is another article I just pulled, The Power of the Placebo Effect. Treating yourself with your mind is possible, but there's more to the placebo effect than positive thinking. Okay, so here we go. Here's an interesting study about a placebo. They often work because people don't know they're getting one. But what happens if you know you're getting a placebo? Okay, here's a study published in Science Translational Medicine, right? They had 3 groups of people. One group took a migraine drug, labeled with the drug's name, so they know they're on the drug. Another took a placebo, labeled placebo, okay? So they know they're on the placebo, and a third group took nothing. So they know they're not on anything, right? And it's interesting, even when they knew they were on placebo, they still had some effectiveness, not as much as being on the drug, but there was effectiveness. And they said simply the simple act of taking a pill or participating in a clinical trial, has a healing effect, okay? And this has been something that has been common in the Duchenne trials, is that the boys in the 6-minute walk test, whether it's Becker or Duchenne muscular dystrophy, that do not get the treatment, whether it's exon skipping or gene therapy, whatever, actually initially in the early stages of the trial show benefit. In fact, sometimes even greater improvement than those that are on the therapy. Okay, and that's actually, now, but the thing to remember is this is measuring certain outcomes. They're not molecular outcomes. They're not direct outcomes of the therapeutic. These are physiological outcomes. You feel better, patient-reported outcomes, trying harder, maybe even physically getting stronger because you're working out more, because you're eating better, right? These are all things that can actually manifest that are real. Okay, you're not going to see the placebo effect, however, if you're measuring gene expression, right? Because, so they take a Duchenne boy that's on an exon skipping therapy that makes dystrophin versus one on a placebo where you don't make dystrophin. They may both walk really well initially, but only one's going to be making dystrophin. And that's the one, and that's going to show you that it's actually the therapeutic is doing what it's supposed to do. In FSHD, if you're designed to knock down DUX4, reachable workspace might show that today you had a better day, or you're in a trial and you're doing better. Actually measuring DUX4 and DUX4 target genes, that'll tell you if it's actually mechanistically doing what it's supposed to do. And then you're not going to have a placebo effect there. Okay, so why do I bring this up? Okay, we're going to talk about 3 things. Okay, first off, there was some news. There's an Epicrispr trial out there. This is CRISPR inhibition technology. I love the concept of CRISPR inhibition. We've talked about the Epicrispr Biotech clinical trial, phase 1/2, enrolling 10 people, 2 different doses with their Epi321. It's a special Cas9 fused DNA methyltransferase designed to shut off expression of the DUX4 gene delivered through AAVrh74 virus at a very low dose that is likely very safe and, in my opinion, very unlikely to be effective. Okay, and so recently this press release came out. It says, Epicrispr reports early clinical activity and favorable safety profile in first in-human epigenetic editing study. Okay, and it says, here we go. So I'm just going to read it to you so there's no question about what's going on. Okay, Epicrispr, a clinical stage company pioneering gene modulating therapies, today reported an early clinical data from its ongoing first in human open label study evaluating Epi321, an investigational epigenetic therapy for facioscapulohumeral muscular dystrophy. Epicrispr holds the first and only open epigenetic editing investigational new drug authorization in the United States, with additional clinical trial application clearances in New Zealand and Australia. The ongoing open label study is primarily designed to evaluate safety and tolerability. That's the key. primary endpoint is safety and tolerability. I predict it's going to be safe. I think most people predict it's based on what we know. Remember, gene therapy has been around for a long time. RH74, the reason they use that virus, has been around for a long time. At the doses they're using and everything that they're doing, it would be surprising to me if this was not safe. You still got to do the safety study. And their primates showed that at 3 to 6 times the dose that they're using, perfectly safe. So we expect safety, but you still got to do it. And so that is the primary endpoint. And then there are secondary endpoints that are exploratory efficacy, right? They're not powered enough to know and properly done enough to know if it's truly efficacious, but maybe you get some indication of efficacy. And these are going to be things like whether they're taking a biopsy before the gene therapy, 3 months, and then 12 months after the gene therapy, and look for repression of DUX4 target genes, methylation of the D4Z4 array, and these types of things. Okay, those are molecular targets. Okay, and there is no placebo and there is no crossover. So everybody in the trial knows they're getting the drug. Okay, so right now they say to date, 3 participants have completed their 3-month follow-up visits. Across these participants, Epicrispr has observed favorable performance, including improvements across a broad range of evaluable strength and functional measures. Performance across these measures generally exceeded outcomes observed in an external comparator cohort, providing early evidence of biological activity. Okay, now this is key. It's not evidence of biological activity. It just could be suggestive of biological activity. They're actually not measuring at this point and not reporting any actual biological activity. What they're saying is that the 3 participants in the trial are doing better than they were before they got in the trial. By some level, things are trending better, okay? But the key is no serious adverse events and no severe adverse events have been reported among the 4 dose participants, as expected, and that's always good news. We always want everything to be safe. Safety is paramount, but you'd like to have safety and efficacy together. So now who are they comparing this to? Well, the external comparator is drawn from RESOLVE, a prospective longitudinal multinational natural history study conducted over 2 years in more than 240 symptomatic adult FSHD patients. Of these, 171 patients meet the enrollment criteria for EPI-321, creating a robust and well-matched comparator arm for contextualizing early clinical interventions. Well, is that really true? Let's think about this. The 3 people that are in the trial know that they are on the drug. They joined the trial with the hope and prayers that this is going to work and save themselves. They are invested. They are not going in as guinea pigs, they may say, but they are going in with the expectation that they are going to get benefit. And again, there's no placebo. They know they are on drug and they really want to get better. Guarantee you that's the truth. How about the people in the RESOLVE study? Everybody in RESOLVE knows that they are not on a drug. They know that they are not on a therapeutic, and they know that they are basically, what is being measured is their decline. That's the whole point of the study, right? We're going to measure how you get worse over 2 years. That's the study. And so what they're saying is that if you compare the people that are in a clinical trial, taking a therapeutic that is hopefully going to make them better, perform better than people who are not in a clinical trial and are not on a drug and know they're not on a drug and know they're supposed to get worse. Okay, that's actually what's being said. Now, does that mean that the drug's not working or the therapeutic? No, not at all. Does it mean it is working? No, not at all. Okay, that's actually, I took it to that. In fact, you could actually look at that and I'd say, you would probably get, I'm willing to bet, the same results if all 3 or all 4 were actually on a placebo. That's exactly the type of outcome you would expect. In fact, you might even, people say I get, people say that, I get a little bit too sciencey. Well, let's get a little bit less sciencey. Let's talk about healing crystals. The science behind effectiveness of healing crystals. Would crystal therapy in FSHD work? Okay. Well, it depends on what you're measuring. If you're measuring DUX4 levels, not at all. But let's say you're just measuring reachable workspace and you're measuring, if you really believe in the healing power of crystals and you're taking your crystal, I've looked online, and you're energizing your crystal in the sun, every day as you're supposed to, maybe it's going to work. So what's the science? Science finds no evidence that crystals heal or alter human energy, and scientific sounding claims like vibrations or the whatever the hell effect don't translate to well-being, you know. But many people still experience real comfort due to the placebo effect, expectation, and the soothing structure of ritual. Used as mindfulness anchors, chosen for personal appeal and tied to simple intention, crystals can help ground attention and reduce stress. Their value lies in the mind habits of users, not in any intrinsic powers, right? So, you know, they have a unique spiritual energy. It could actually be real if you really believe in them. So maybe we should have a clinical trial on crystals. We'd end up with the same press release. Okay? Potentially. Now, again, that doesn't mean it's not working, but what you need to see, in fact, you know, it would be actually shocking if you didn't get these results. So why would someone put out these results? I mean, what you want to see is DUX4 repression. And after 3 months, I would expect full DUX4 repression. AAV is fully expressed by that time. You're not going to get any really more expression. You should have target engagement. They should have methylation of the D4Z4, and they should have full, if it's working, it should be working full boat by 3 months. You wouldn't expect it 12 months to, you expect it to still be working. I guess for the concern at 12 months would be immune clearance of CRISPR containing cells. We talked about that in the previous podcast. You know, so maybe, but it's not likely to get better. It's more, if anything, it's more likely to go down. So at 3 months, you should be able to see that. So why is there a press release here? Well, you know, remember, these are for investors. This was presented at JP Morgan Healthcare Conference this week, and this is for investors, to hope that investors are as uninquisitive as the guys at Solve FSHD. Okay. But anyway, that's enough about that. I just wanted to kind of explain this because I've seen a lot of things. Again, it's not bad news. It's not good. And to me, that, you know, it's safe. That's the key, right? It's safe. They'll do it properly powered. I have no doubt that they will find this to be a success as a safe thing. And then these likely unefficacious doses will go forward into a phase 3 trial if they can get enough for someone to pay for it. I guess they're really going to have to sweet talk Chip to get someone to pay for it. Maybe you will. All right, so that's Epicrispr. We still wish them the best. I just wish they had done a better job. I wish they'd used a better next-gen virus, and I wish their promoter didn't look so wonky in their preclinical data. You can look at my previous podcast and understand what I'm talking about. All right, let's move on, and let's talk about the RESTEM trial. Okay, RESTEM announces a first patient dosed in the phase 1/2A study of RESTEM-L in FSHD. This is back from December. And this is being done at Stanford. John Day is leading this, Dr. Day, fantastic neuromuscular disease researcher, one of the best in the world. And I've talked to him about this trial. He's like, you know, I thought it might actually have some real potential. And which is, you know, it's interesting because if my memory serves me correct, he was not willing to participate in the phase 3 Fulcrum nosmapimod trial because as we understood, that was not likely to have any benefit. But he's participating in this. So, you know, it's always good when the people driving it believe in it. So what's the deal with this? It's stem cells, okay? Let's read again from their press release. These are written for the general audience, so I figure it'll save me from being too sciencey. Okay, RESTEM is a clinical stage biotech company that develops off-the-shelf next-generation cell therapies designed to modulate the immune system. Today, they announced the initiation of RESTEM-L, the company's umbilical lining modified progenitor cells off-the-shelf platform for the treatment of FSHD. And this is funded by Solve FSHD. You know who those guys are. Okay, so what is this trial? It is a double-blind, randomized, dose-repeating, placebo-controlled crossover study to assess the safety and preliminary efficacy of allogenic UMPCs on disease severity in patients with FSHD. Okay, this is led by John Day at Stanford. They're going to do 16 patients. So 1-to-1, so it's placebo. So you're going to get either RESTEM-L, the stem cells, or the placebo for 6 months, and then you're going to switch. Okay, so that's kind of interesting because you don't know whether you're on it or not. You know that you're not going to be on it for 6 months and you are going to be on it for 6 months. It's a 12-month study and the endpoint is safety. Again, phase 1/2, safety. I'm likely safe. My scientific opinion is that this is not the first time these have been used in people. So think they have a good safety profile. And again, they're going to have exploratory outcomes looking at inflammation. So why are they looking about inflammation? Well, basically, what these things are, so these are stem cells. And everyone hears stem cells and you think you're going to be rebuilding your muscle. Okay. Again, there's different types of stem cells. This is not a case of muscle stem cells that are going to fuse with your muscle and build back muscle. This is not cell therapy in that way. What these essentially are is, you'll see it right here, okay? The UMPCs are stem cells. They modulate key inflammatory factors in muscle inflammatory indications, okay? So basically, what these things are is they are little cells that are going to be pumping out anti-inflammatories, cytokines, growth factors that try to improve muscle health, try to tamp down the immune response that we know is activated in FSHD, and in that way slow down disease progression and pathology. But they are not treating, so the Epicrispr trial and the is actually trying to shut off DUX4 at the gene. Avidity, Delbrax, that is shutting off DUX4 by eliminating the DUX4 mRNA. Okay, so those are both getting at kind of the key driver of pathology in FSHD. What this trial is doing is DUX4 is going to be going, cells are going to be dying, but you're going to prevent the immune response, or that's the goal, is to tamp down the immune response to delay or slow down or maybe even stop progression of some pathology that is mediated by the immune system in response to DUX4 expression, DUX4 target gene expression, and in FSHD muscle. Okay, so basically, you're taking anti-inflammatories. That's basically what's going on here. Okay, of course, they're going to be in your system for a while, and they're going to be continually pumping it out. And, we'll see. I hope there's some benefit here. I can conceptually see how you could have some benefit here. But in the end, again, it's the type of thing I would rather see coupled with something like Delbrax, or if Dyne ever gets in the game, or Arrowhead, or one of these antisenses or something, to kind of tamp down pathology while you're shutting off the initiating event. But whatever, maybe there's some benefit in anti-inflammatories. Some of the nutritional things that people do are to, deregulate, or not downregulate your inflammation, your inflammasomes and stuff. But that's essentially what this is, okay? 16 people, number of people have asked me, I never like to say yes or no to a trial. I guess I shouldn't say that. There are some that I say yes, but I never say, but it's a trial where, fundamentally, I understand the basis of it. Fundamentally, there's a scientific rationale for doing this in FSHD. Are you going to see, you're going to, long-term benefit? In the end, you're still making DUX4. And in my opinion, the muscles are still going to degrade. Maybe it'll slow it down. And, if have the disease, you'll take whatever you can get, right? But that's basically what's going on there. One thing I will give the company credit, though, there's always the temptation to set up shop in Mexico or Honduras or China or Korea or somewhere and have the rich people go over there to get injected. Basically what Saul said to them is, let's run, if it's real, if we're going to really see benefit, do an actual clinical trial in the US. And we'll see if this actually works. Because the problem with all these stem cell treatments that people do is they're uncontrolled, they're all around the world, and you don't actually have people that understand anything about FSHD measuring anything. They are patient-reported outcomes, and guess what? We just talked with placebo. Patients always report that they're doing better, but are they? Yeah, who knows, right? At least here we'll get an answer. So actually, I'm glad that they're doing this trial, and I'm glad John Day is doing it, because you can't get any better than Dr. Day. And, great, Tina Duong's over there, and she's fantastic, part of the team there. Actually, the whole Stanford team's fantastic. And, it's not my favorite therapy, but hey, let's see if it works. Let's see if there's any benefit, right? Because at this point, right, you'll take anything that gives you benefit. So we'll actually find out on some level. Of course, this first one is safety. It's only 16 people. It's only going to be indicative of going to phase 3, where you have a larger trial that can actually show that. But there should be some indications from this people, especially with the crossover placebo-controlled setup, that you're getting benefit. But we'll see. We'll see. All right. Then most recently, Scholar Rock. Scholar Rock, you guys maybe no one in the space ever heard of that. I wasn't sure if we were allowed to talk about them or not. Scholar Rock is a really cool company. I know several of the people involved with that. I've met them at several of our Shark Tank meetings. And this kind of came out from, Neil Camarda has set up, from FSHD Canada, has set up these kind of meetings where to understand what Neil and Chip and the gang want is they understand that we're well on our way to shutting down DUX4 expression. And trust me, we're not there yet. Even if Avidity gets approved, I guarantee we can do better. But anyway, you can conceptually see that we're going to be able to shut down, as in the space, DUX4 expression one way or another, at least decrease its level of expression. There's a lot of variables in that. But we got to get our muscles back. If all decreasing DUX4 does for some of you is stop the destruction, and unless you're very mildly affected, most of you aren't going to get your muscles back. All right? You're not going to suddenly take Avidity and get up out of your wheelchair and go dancing. You're just not going to get any worse. Now, some of you are going to get better depending on who you are and how your muscles, and we don't fully know where these things are going to be, but chances are you're going to need a second little helper, a boost, whether it's muscle, whether it's cell therapy or small molecules that are going to drive regeneration or something along those lines, and that's what you're going to need in order to get going, right? And so what's the deal? What is Scholar Rock? Well, Scholar Rock is playing around with the myostatin pathway. You've heard about this a lot, right? Myostatin stops muscle growth. Okay, so blocking myostatin would then stimulate muscle growth. Okay, follistatin treatment would stimulate muscle growth by inhibiting myostatin. There are anti-myostatin antibodies. If you remember the Acceleron trial, right, when Acceleron had ACE83, they had a localized myostatin inhibitor that they were injected into biceps. The muscles got bigger but not stronger, and so it didn't really provide you any benefit. But this is a pathway that for many years, many groups and companies have been trying to play with, right? Trying to modify. Now the problem is that the pathway affects many different processes in cells, and so can you actually get the benefits without losing the things that you need if you block the pathway? Okay, so there's so many points of intervention in these pathways that everybody's kind of approach is slightly different. And there's this idea that, well, if you just do it right, you'll be able to increase muscle mass, increase muscle strength without getting the bad things that can happen with blocking the whole pathway. Okay, now keep in mind that Roche has a myostatin inhibitor trial that actually we're going to probably hear the results this summer. Some of you may have been in that. Again, Acceleron had a trial, two trials actually, back maybe 5 years ago. So this kind of approach has been around. But Scholar Rock, so conceptually, it's the same sort of pathway, but devil's in the details. And so they have this thing, they've been working on this thing called Epicrispr, and the MAB is for monoclonal antibody. And what this is, it's a fully human monoclonal antibody that inhibits the activation of myostatin. So you're going to block myostatin. It's a negative regulator of muscle mass by selectively binding with high affinity to pro-myostatin and latent myostatin. Okay, so they're going to be a little bit different than the other therapies that are out there. Going to basically block myostatin and pro-myostatin. So maybe they're going to do a better job. That pro-myostatin is a preprocessed form of myostatin. So maybe they're going to do a better job blocking myostatin and block the pathway. So why do we care? Because well, they've been working in the spinal muscular atrophy space. Okay, SMA. Okay, now SMA is it's fundamentally different because that's going to be motor neuron degeneration, but you actually get muscle fiber atrophy. Okay, and so what's happening is as patients are getting treated and for SMA, so the SMN targeted therapies, whether it's gene therapy or antisense therapy to restore SMN function, now they're left with muscle, so the nerves, it's getting fixed, but they almost have a second disease because they developed and they have all this muscle atrophy. Now we got to get the muscle back, get the muscle strength back, get the muscle mass back. And so actually in clinical trials, it turns out the treatment with Scholar Rock has actually been pretty good. It actually has been pretty good, pretty effective. Okay. And so one of the things that in humans, in clinical trials, in combination with SMN targeted therapies, okay? And so one of the things that Neil Camardo did, it was a brilliant move, was when thinking about how to address this with FSHD, he said, well, let's talk to the people at the SMA Foundation. Okay, and the key person there is Karen Chen, just absolutely brilliant, fantastic person running the science at the SMA Foundation. And she's kind of come on board with Neil's project for FSHD. And the idea is that things that are good for SMA, maybe they'd be good for FSHD. And conversely, what she's getting out of it is maybe some things that we find good for building back muscle in FSHD, maybe they could be used for SMA, right? So it's a good collaboration thing, makes a lot of sense, the same kind of therapeutics. Now fundamentally, though, the diseases are different. Right, SMA is a motor neuron disease, FSHD is skeletal muscle disease. But once they're kind of cured, you have the same problem. You got to get your muscle mass and strength back, okay? So Neil and the gang have been working with Scholar Rock to get them to understand the FSHD space. Actually, that's one of my roles has been to help them understand the FSHD space and evaluate the potential for this in FSHD. And so what did they do? They've come out and said, actually, at the same meeting, at the JP Morgan meeting, that they're going to actually do a apitegromab IND application. It's actually been cleared to do a phase 2 study in FSHD in mid-2026. Okay, now I'm not sure of all the details on this. Now I tell you, I found something. This was on the FSHD Global website, and it's not completely right, but it was kind of interesting. I couldn't find where they got the information. But basically, they say they've announced that phase 2 FORGE clinical trial is on track to initiate mid-2026. That's true. They're developing for the treatment of people actually with SMA, and so now they're going to use it for FSHD. They do have already cleared the IND, and they're going to do randomized, double-blind, placebo-controlled trial. The thing that's not quite right is says this is the first IND cleared for a muscle regeneration approach in FSHD. Again, Roche is already actually in trial. Acceleron has been in trial. There have been trials, numerous trials in this space, so that's not quite right. But that's irrelevant. It doesn't matter. It's details. There's just hopefully, and again, I can't wait to see the results from the Roche trial to see if there was any benefit. But again, it's completely placebo-controlled. But what they're going to be measuring now, the key is just like with the RESTEM trial. Okay, they're not addressing the fundamental cause of pathology. This will have no effect on DUX4, right? So that actually becomes kind of the question. I've heard of this for several years actually, and I've always thought this would be a great combinational therapy, right? In fact, I think most of these muscle regeneration things are great combinations. Stop the destruction, build back the muscle. Now, so it looks like they're going to maybe jump the gun here and just say, can we actually get some benefit without stopping DUX4? Now, the caveat to that, in my opinion, is that that's going to be very dependent on the level of severity. And by the way, I'm not advising them on this at all. So I have no connection to the company. I'm familiar with the space. I've seen the presentations. I'm not an advisor at all. I'm not an advisor for Solve. I have no connection to actually any of these. So if you're very mildly affected and you have most of your muscle, you're probably going to see the most benefit. Now, will you notice the most benefit? You have, your therapeutic window might be smaller. You already have the most range of motion, so you get even more range of motion. If you don't have much muscle at all, I don't see how this is going to be very effective, because it's basically you're going to block myostatin. But if all you have is fat and fibrosis, that's maybe not going to work so well, right? So it's going to be really important who you pick to be in the trial if you're not going to shut down DUX4. Now, even if you are going to shut down DUX4, right? So it'll be very curious how this goes. But conceptually, again, it's just one of those things where the idea of inhibiting myostatin, increasing muscle mass, you think, God, this has to work. The history of it has been, it hasn't worked that well in FSHD. But again, the devil's in the details, different molecules, different parts of the pathways, different levels of inhibition, different side effects. All these things are kind of different. Scholar Rock's molecule looks pretty good in SMA. And I'm not sure what they're sharing with their preclinical work. I'm not at liberty to say anything, but I'm enthusiastic about this as a combinational therapy. And I hope it works as a standalone, at least again, if we're talking about, putting the finger in the dike, so to speak, slowing down progression, bailing the boat out faster while it's sinking, you'll take that, right? You'll take what you can. So, in some ways it's similar to the RESTEM trial. It's not attacking the root cause of the disease, but hey, there is a scientific basis for this to go. In fact, there's a scientific basis for all three of these, right? So, there's good stuff going on in the space. Let me just kind of kill the share. So that's kind of what's going on. I don't know if you like the new format. So I'm trying to be a little bit shorter. But that's kind of my take on what's going on. I'm pulling stuff off the web, reading directly from their sites and giving you my real-time take on everything. So, in summary, the Epicrispr news release, glad to hear that it's not having any adverse effects and it's as safe as we expected it to be. I don't think we can read anything into whether biological activity. That's for investors and to hopefully convince them. I'll be looking forward to hearing about DUX4 target gene repression. Keep in mind that with an N of 3, statistically, one of them is going to go up, one's going to stay the same, and one's going to go down if there's no effect. If it's working, all three of them should really have target gene expression go down and should see significant levels of methylation, although it's hard to say. I don't really care if there's methylation or not, as long as the target genes go down. I hope it works. Wish them the best. I just conceptually, boy, I wish they were using a next generation vector and a little bit higher dose to really hit all the cells and give it a real chance at efficacy, in my opinion. And I hope I am proven wrong. The RESTEM, that is immunosuppression mediated by these kind of progenitor cells isolated that are kind of engineered and isolated from umbilical cords. They are not rebuilding your muscle. They are tamping down the immune response, producing beneficial factors. They really can't tell you exactly what they're producing, but things that are beneficial to muscle health and hopefully will prevent the immune system from responding to DUX4 and driving any DUX4 mediated pathology. That's the hope. Being done at Stanford. And then, of course, Scholar Rock, this is myostatin inhibition, a unique molecule that has shown benefit with SMA in patients at improving muscle health after therapy. So a combinatorial thing, restore SMN production, and then restore motor neuron health, and then helps build back some muscle. In FSHD, the idea is going to be that you're building up muscle, keeping in mind that you're not, at this point, affecting DUX4 expression and driving a pathology. But as a combinatorial, maybe in the future, I think there's some legs to it. I'd be curious, it would be an interesting thing combinatorial to take forward with whatever does stop DUX4. Let's stop the destruction and build muscle back. All right, well, that's kind of my, I'm going to kind of try to keep this format a little bit, try to make it hopefully more understandable for everybody. And feel free to send me any questions, peterjones@med.unr.edu. Happy to do my best. Sometimes it's taking me longer and longer to get back to everybody because we are overwhelmed with our samples for our epigenetic study and our research testing and developing our own therapeutic lines. And, the space is moving, space is moving. There's a lot going on, and I think 2026, you're going to see a number of opportunities to participate in clinical trials. And I got to some are going to be, in my opinion, some are going to be better than others, more worthwhile. I just want you to understand the science so that you can make an informed decision for yourself. One additional thing I guess I would say about I expect all three of these to be, the phase 1 to remember is safety first. Okay, that's safety first, with some secondary outcome measures that are going to suggest efficacy. The phase 3 trials are truly where we're going to look at efficacy. And, I just keep our fingers crossed. And, I got to tell you, I got some inside information. There's some really good stuff coming too. So it's going to be a good year. Zahara, I'm telling you, we're coming. It's getting there. All right, Maggie, everybody out there. By the way, thank you for the Christmas cards to all my FSHD young adults and adolescents and things. I really appreciate it. Really, things are moving. I know it's going too slow, but it's getting there. We're getting there. And, hell, if Arsenal can win the Premier League this year, we can cure FSHD. I'm not sure which one's more likely, actually. I think FSHD is more likely, curing FSHD. All right. We'll catch y'all later. Jagger, my man, take us out of here.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. Do you have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information.